Hello everyone, welcome to Get My Class. So let's solve this question, which is identify the lane 1, 2, 3 and 4. And they have given gel electrophorus picture, and they have asked, they have given the options like lane 1 is undigested, lane 2 digested, lane 3 undigested, lane 4 digested. Option B, lane 1 undigested, lane 2 digested, lane 3 undigested, lane 4 digested. And option C, lane 1 digested, lane 2 undigested, lane 3 digested, lane 4 undigested. And option D, lane 1 undigested, lane 2 digested, lane 3 digested, lane 4 digested. So let us understand the question. It says identify lane 1, lane 2, lane 3 and lane 4. Okay, you have to identify these lanes. Like this is the lane 1, this is lane 2, this is lane 4 or 3, lane 4. So first of all, what is this? This is a method of isolation of DNA fragments, and this method is called as gel electrophoresis. What is this? Gel electrophoresis. Where on gel, this is the agar gel, on gel, they use electric current and they try to separate the fragments. Okay, this is separation of fragments, DNA fragments here. DNA fragments is done. And since the DNA is negatively charged, the DNA will move from cathode to anode. Cathode to anode. Cathode is negatively charged, and anode is positively charged. And we already know that DNA is what? DNA is negatively charged, and hence it will move from cathode to anode. So while moving, based on their size and molecular weight, they get separated. Okay. And if it is undigested, it will not move further. Okay. So you can see that in lane 1, the DNA is not digested. It is undigested. So it is not getting separated as fragments because it is one whole thing. And that is why it is undigested. What lane 1 is? So lane 1 is what? Undigested. What is digested means? Like treating it with restriction enzyme. When you treat with restriction enzyme, it will break or it will cut the DNA into fragments. So lane 1 is undigested. Lane 2 is, of course, it is digested because you can find fragments. Okay. So lane 2 is what? Digested. Digested means treated with restriction enzymes. And lane 3, you can see the various bands, and lane 3 is also digested. Okay, lane 4, you have again digested because you can see several bands. So this can be considered as a ladder also, either ladder. Ladder is the, you can say, reference point. Okay, where for reference you will know which are the fragment on the other lanes. Okay, or you can simply just say that this is also digested because you can find the fragments which are largest and smallest. So lane 4 is also digested because it's been treated with restriction enzyme, and hence you will find various bands. So let us see. Option A says lane 1 undigested, that is correct. Lane 2 digested, that is also correct. Lane 3 undigested, which is incorrect because lane 3 also digested. Hence it is ruled out. Option B, lane 1 undigested, correct. Option that is lane 2 is digested, correct. Lane 3 again undigested. So this is also ruled out. Option C, lane 1 digested, lane 2 undigested. Lane 3 digested. So lane 2 undigested they say. So here itself you can just cross it off. Option D, lane 1 undigested, correct. Lane 2 digested, correct. Lane 3 digested, correct. Lane 4 digested. Yes, that is the correct option because as per our description, you can find fragments only in the digested lane. So 2, 3, 4, you can find digested lane. And lane 1 is undigested. So hence the option, which option is correct? Option D. I hope you understood the explanation. Thank you, everyone.